The next type of computation, parallel computation, is one that's loosely coupled, meaning there are phases of independence followed by phases where they have to work together. So the schematic diagram is we're going to have 4 input files. There's going to be some initial computation on those input files. Then those workers are going to communicate with each other. That's going to create a set of 4 different independent tasks in the back end that will then have their different output files. So this fits really nicely into that MapReduce paradigm, where up front you're going to have our mappers. They're going to shuffle to communicate with each other, and then a series of reduce to produce our final results. That's really the special sauce, if you will, of MapReduce about why it's more powerful than, say, Condor or Grid Engine, is that it supports this shuffle over very, very large data sets. So you can, again, if you think about this sort of conceptual model, it's sort of like, we're going to choreograph this dance where everybody has a dance partner, then we're going to blow a whistle, everybody's going to find a new dance partner, and then continue on. So this is extremely important for genomics, and it fits really well with many, many applications that we may be interested in. So in particular, there's this computation that I alluded to before called short read map. And the idea is we're going to have our genome, and here I'm just showing a shot of a few, couple of dozen nucleotides, whereas in the real genome we're going to have about 3 billion nucleotides. Many, many billions, very short read. Here, I'm just showing 10 bases, but in reality it's about 100 bases. We're going to take out billions of these 100 base pair short reads, try to line them up to our genome to identify variations. So here we have the reference genome as a C, but all the reads that line up there are an A. There's some really nice algorithms, some really nice string matching algorithms that are very efficient for taking a single read, mapping it onto the reference genome. But as efficient as it is, it still takes on the order of 1,000 CPU hours just to analyze one genome. We're currently underway doing thousands and thousands of genomes, so that translates into millions of CPU hours to be able to do this type of analysis for one project. So it's just going to take way too long to try to do this on one computer. We're going to want to spread this out over many computers. In addition to this computation trying to identify where the variants are, this sort of read mapping and coverage analysis is really fundamental for many different assays. Studying, complex mutations in the genome, studying the biologically active regions, that's called RNA-seq, studying where proteins are binding to the genome, that's called ChIP-seq, studying different modifications to the genome, that's called methyl-seq, and then studying how the genome actually folds up on itself, that's called Hi-C-seq. So here I'm just going to be talking about, finding variations, but this is really, really fundamental to computational biology and extremely important for a number of applications. So the nice part is this fits really well into a MapReduce framework, and in particular we were able to use a feature of Hadoop called this Hadoop streaming that let us tackle this problem in a very efficient way. So the goal is to align billions of short reads to the reference genome to identify single nucleotide polymorphisms where the SNPs are, and we're going to be able to reuse as much software as possible using this Hadoop streaming. So the input to this new pipeline that we call Crossbow is going to be some set of those sequence files. Each of these files is going to have millions and millions of reads in it, and there's going to be here I'm showing 4 files, but it could be thousands of these files with tons and tons of biological sequence data in it. The first step is going to be to run this sequence alignment algorithm called Bowtie that was developed by my collaborator Ben Langmead. And the idea is that Bowtie knows how to take a single one of these reads and then quickly do string matching to find where it best aligns to the reference genome. So normally Bowtie is run, one copy of Bowtie per computer, and what I'm trying to show here is that we're going to launch 4 copies of Bowtie on 4 different computers. This could be 400 different copies of Bowtie on 400 different computers. And then we're going to take the output of Bowtie and then emit it as a key-value pair, where the key is going to be the chromosome, what we call the chromosome region that the read aligns to, and then the value is going to be the information about that alignment. So your genome is organized into 23 chromosomes, So that will allow us to have some nice natural partitioning. So the idea is that the reads are going to align more or less randomly to these different chromosomes, but then we're going to tag where those alignments are so that we can tag all the alignments to chromosome 1, tag all the alignments to chromosome 2, all the alignments to chromosome 3, and so forth. Actually, in reality, we're going to do this a little bit better because we're going to tag it and be like, oh, we're in the first 10 million bases of chromosome 1, the second 10 million bases, the third 10 million, the fourth 10 million. That'll give us better opportunity for load balancing down the road. Given those key value pairs of where the alignments occur, we're going to use that Hadoop shuffle functionality to collect all of the alignments to those different chromosome regions. So here's all the alignments being collected to that first few megabases of chromosome 1, the second few megabases, the third, and so forth, all the way across all of the chromosomes. Those are going to get sorted and shuffled into place across the different machines. And then for our reducer, we're going to use another program called SoapSnip that knows how to scan through those alignments and identify where those polymorphisms. It has a really nice model for discarding where the sequencing error occur. So we built this pipeline using a small cluster at the University of Maryland, but in our main publication, we scaled this up to run on the Amazon cloud in EC2. Now, it does take time, and also it costs money to transfer data in and out of the Amazon cloud, but in our case, it wasn't so bad. So our input data was 3.3 billion short reads, 35 base pair reads. It took about an hour and 15 minutes to do the transfer. It was about 106 gigabytes of data to do so. The trick for doing this was to, rather than just using like FTP in one file at a time, we were able to FTP 40 different files at a time. I should have said that the data were housed in a repository in England, and then we were transferring it to Amazon in someplace in Northern Virginia. So we're able to do that transfer very, very efficiently by executing 40 parallel FTPs at once. There are actually better transfer mechanisms for doing large data transfers. There's something called Grid FTP, and there's a commercial client called Aspera that can just make better use of the bandwidth available than just a regular FTP. Nevertheless, it only took us about an hour and 15 minutes. At the time that we did the transfer, it cost us about $14 to do the transfer, which, yes, is money, but relative to the thousands of dollars it took to generate these data, $14 to transfer is just a tiny fraction of the data. And then in our main experiment, we ran our Crossbow pipeline on a cluster rented from Amazon EC2 with 320 cores in it. So this was 40 computers with 8 cores each. It takes a little bit of time just to set up a cluster of that size. It takes some time to run many copies, in this case 320 copies of Bowtie. And then it takes about an hour to do all the variation calling, but not so much time. So end to end, including the time to do the data transfer, it took just around 4 hours. At the time that we did the experiment, it cost about $100 to do the experiment. If you were to reproduce this exact experiment today, Amazon has gotten quite a bit cheaper, so it would be about $50 or $60 to do this transfer today. So we think this is a really compelling example of how cloud computing and MapReduce can be used in the life sciences. Because we started with 3.3 billion reads, we were able to very rapidly transfer the data, do all the computation to identify about 3 million of those single nucleotide polymorphisms in an afternoon and for less than $100. So it was really a compelling example of how these technologies may be used. Since that publication, we recognize that this sort of framework of mapping reads to a reference genome, shuffling them into chromosome regions, and then scanning those alignments occurs all the time under many different contexts. And we've been developing a new system that we call Jnomics, that rather than focusing on just Bowtie and just SoapSnip, is going to support many, many different genomic sequence analysis tools to be able to do it at a very, very large scale. So far, we've been using these technologies to study human diseases like cancer to see how your genomes are mutated, and then also different plant systems to try to figure out why different strains of rice and different strains of corn and different strains of wheat are able to grow with different properties, like how fast they grow, how tall they get, how big the fruits are, and so forth.